Howdy, folks. Thanks for checking in to Mr. Ulrich's landofbiology.com. I am Mr. Ulrich. In this installment, we're going to be looking at the organelles of eukaryotic cells. And as a family album, yes, there will be pictures. You're going to get a chance to actually see them. I'm going to include both micrographs, the pictures of the structures themselves, as well as some diagrams so that you can see what they're going to look like on like a test or in a textbook or something like that. Now, of course, we should take a moment to explain what a eukaryote is in comparison to a prokaryote. Remember, you are a eukaryote. Your cells have nuclei. Pro rhymes with no. Bacteria have no nucleus. Now, the nucleus is one of the membrane-bound organelles that makes a eukaryote. But that's the deal. It's those membrane-bound organelles. Let's start with that prominent organelle, the nucleus, the one that you're going to see right off the bat when you look at a cell, even under our compound light microscopes that we have in class. When you draw the fried egg diagram of the cell, it's the yolk, and you can put that right in your notes. When we look at an actual cell, here's a phase contrast microscope view of an amoeba called Chaos. Chaos, fabulous name. It is that prominent structure in the middle. Now, a lot of students come to me from middle school being able to label the nucleus on the diagram. Yes, that Pac-Man looking thing in the diagram that you have. But they'll tell me that the nucleus of the cell is the control center. It tells the cell what to do, which is a little bit misleading. Remember I was saying organelles, what they are compartments. What the nucleus is it's the compartment for DNA. See, the cytoplasm, the part outside of the nucleus, is kind of a dangerous place for nucleic acids like DNA. And so having it inside of a nucleus protects the DNA. Now, DNA, of course, is the instructions for protein construction. So if we were thinking of a principal life function for the nucleus, it would be synthesis for those proteins. Within the nucleus itself are some tiny structures, sometimes there's more than one, called the nucleolus or nucleoli if there are more than one. The little old nucleus. If you take nucleus and you put a little O in the middle of nucleus, you get nucleolus. Anyway, the job of the nucleolus is to produce a different nucleic acid called RNA. The RNA has a lot of different functions that we'll talk about more when we get into molecular genetics. But RNA also makes up the ribosome, which, as we're going to find, if you haven't taken reproduction and development already, we're going to find RNA is involved also in protein synthesis. So again, its life function is going to be synthesis. Surrounding the nucleus is a maze-like network of membranes called the endoplasmic reticulum. It does come in a couple of varieties. It doesn't matter which kind we're talking about. Here's the smooth endoplasmic reticulum. Most of the time we're talking about rough endoplasmic reticulum. What makes them rough are ribosomes. We'll talk about ribosomes in a second. But the endoplasmic reticulum is really just a folds of membranes. And these membranes make the little compartments for making proteins and allowing proteins to complex with other proteins and make really neat big proteins and all kinds of cool stuff like this. Regardless of whether it's smooth or rough endoplasmic reticulum, both of them are involved, again, in this process of synthesis. I hope you're noticing a trend in these life processes. So far, we've been talking about organelles that are made out of compartments of membranes. The ribosome actually is not a membrane-bound organelle. We're going to find ribosomes within all cells, including prokaryotes. Ribosomes are incredibly important. They are the actual site of protein synthesis. So this is where different amino acids get assembled together in a chain to make particular proteins. They are the little tiny berries here. This is endoplasmic reticulum. This is a tube of endoplasmic reticulum kind of pointing out at us. And the little berries around the outside are ribosomes. They're going to be the little dots that you see on a diagram. Oftentimes they're associated with the endoplasmic reticulum, but not always. They are also floating around in the cytoplasm itself. Again, since they're involved with protein synthesis, their life process is going to be synthesis. There's another network of membranes folded back and forth, making little compartments called the Golgi bodies or the Golgi apparatus. They're named after Dr. Camillo Golgi, who he actually came up with a staining technique called black staining or dark staining for staining nerve cells. This was in the late 19th century, 1870s. And one of the things he noticed when he was staining these cells were the structures within the cells. And he started to hypothesize about organelles and internal structure of the cells. And he wasn't really recognized as being accurate, but pretty visionary. That's why they're called Golgi bodies. But anyway, these structures are where proteins actually come to become part of a membrane or to become associated with a lysosome. So this is lysosomes are structures that contain digestive enzymes. This is also where proteins that are going to be secreted outside of cells or become part of the cell membrane are kind of packaged for secretion or for incorporation with the cell membrane. Now, they're not really making things here. They're kind of the final assembly and the packaging department. We think of them almost as like the UPS or Postal Service. And so their principal life function is going to be transport. Lysosomes, as I mentioned before, are structures that are actually made at the Golgi apparatus or Golgi bodies. The edges of the Golgi bodies kind of pinch off into little sacs, little vesicles, and inside of those sacs are going to be, in this case for lysosomes, digestive enzymes. The prefix lys, like lysol, means to break things down. And so these are bodies, that's some, that contain digestive enzymes. In this regard, they kind of work for nutrition. They allow the cell to take in raw materials so that it can absorb them and use them for other metabolic processes. Now, in a diagram, it becomes really hard to tell a lysosome from any other vacuole. A vacuole is just a general term for a vesicle for storage. If they have water in them, then they're water vacuoles. If they have food in them, food vacuoles. Waste, it's waste vacuoles, etc. If they contain digestive enzymes, then they are considered lysosomes. So these vacuoles store all kinds of different stuff. They are defined by what they contain. What the vacuoles allow the cell to do is actually move these different materials around, water and wastes and all these different materials where they need to go throughout the cell. Another very important membrane-bound organelle are the mitochondria. In your diagram, they alternatively look like a slipper that has like mustard in it, or a hot dog that's been cut in half and also has mustard on it, I guess. Really what it is it is a large bag inside of a small bag, and so that large bag gets folded as it's inside of the small bag, and that creates lots of surface area. It also creates a compartment within a compartment. We'll get more into the structure of the mitochondria and how it works in AP biology, but for us right now, we just need to know the mitochondria, yes, is the powerhouse of the cell, in that this is where most of the energy is actually squeezed out of those glucose molecules and converted into ATP, the very important molecule that is going to kind of power a lot of the cell's machinery, active transport and movement and stuff like this. So their principal life process is going to be cellular respiration. Again, we're not talking about breathing, all right? What we're doing is we're talking about releasing energy or making energy available, transferring energy from food molecules to ATP so that it can be used by the cell. Another structure that is also a cell kind of within a cell, it has its own DNA, just like a mitochondria does, is called the chloroplast. Chlora is for green. If we look at our animal cell diagram, you might notice there isn't anything that looks like a chloroplast, because of course these are only found in plant cells. This is the site of photosynthesis where glucose is synthesized from carbon dioxide and water. Because of this is going to be the life process of synthesis. Cytoplasm is not really an organelle. it gets kind of short shrift when we're talking about cellular structures, and I think that's because cytoplasm doesn't really have any structure at all. It's that watery goo between the nucleus and the cell membrane, and a lot of times it gets left at that. But it is a real complex goo. It's a very complex stew. That's where all the cellular activities are going to be taking place. All the reactants, all the raw materials for all those chemical reactions, all of the enzymes, all of the products of those chemical reactions, all the modifiers, everything, all that water. There's a lot going on in the cytoplasm. When we start talking about all of those life processes or all of those chemical reactions, the word that should come to mind is metabolism, since that kind of includes all of the cellular activities of the cell. That brings us to the cell membrane, that outermost structure of a typical animal cell or sarcodeme. The cell membrane is very important. Please remember all cells have a cell membrane, even if they have a cell wall like a plant cell, or like a fungus, or like most protists, or like a bacteria, or like most cells really when you think about it. They all are going to have a cell membrane. Sometimes it's called the plasma membrane. New York State Board of Regents likes to refer to it as the plasma membrane. Plasma kind of makes it sound like it's like a fluid, and that's good because the cell membrane does have flow. Another term for it describes its structure. It's called the lipid bilayer. This is a structure that you're not actually going to see when you see a diagram. It's really going to make the boundary between where the cell stops and the external environment begins. And it's really only a couple molecules thick, so you don't actually see it, but you see where the environment kind of changes in that regard. When we look at the actual pictures themselves, you can see the two layers, the lipid bilayer. I really like this diagram over here. Let me get my pointer. I really like this diagram on this side, where you can see the real different structure over on this side, much more shaded in from this side over here of the cell. And that's really what the cell membrane does. Is it's going to maintain a different, a stable internal environment, even though the external environment can change. And it's going to do that by allowing certain things to go through. Now, of course, before you write down maintain stable internal environment, we know a stable internal environment is homeostasis. So you can just write it maintains homeostasis. It's going to do this by controlling what materials actually pass through that cell membrane. We're going to get more into cell membranes and their structure and their function later on. For right now, we need to recognize that they are the outer boundary of the living boundary of the cell, animal cells especially, before the cell wall, if there is one, and that their principal life process is regulation, since they are involved in maintaining homeostasis, keeping that inside normal when the outside changes. That'll bring us to our very last cellular structure called the cell wall. And we've actually been looking at the cell wall the whole time. There it is in the background. Right, these are plant cells, and the rigid outer structure of the plant cell is made of a cellulose cell wall. It's actually what you're writing on right now. That's what makes up paper. All right, here is Robert Koch's first view of cell walls from Sliced Cork, where he described the cell wall as that structure. It is non-living. So when the cell dies, like this cork is dead, but the cell wall kind of stays behind like wood or paper, since those are dead cells. The cell wall is what actually holds the plant up against gravity. So it doesn't really protect the cell so much. A lot of students will tell me that it protects the cell, but really its job is to keep the whole plant upright, really so that it can get closer to the sun and get the light that it needs. Okay, I think that's enough for now. Thanks for checking in. Again, I am Mr. Ulrich, and if you have any questions, feel free to drop me an email. Other than that, I'll see you in class.